Hello, my name is Annie Pham, and I am in Bio 105, Section 1. This is my Exam 1 regrade. Yes. Okay, so question 1 is basically asking if Jup 1 would be a good genetic model of development or not. And I'm going to change my answer from what I said, and I would say no. I disagree because of these following reasons. In order for it to be a good genetic model, it would have to have good insertion and deletion. And here, Jup1 actually has really bad DNA repair. It just means that it's constant damaging, which means that it takes away from development time because it has to be fixed over and over. And it doesn't have much mutation or DNA aberrations. So I would say that Jup1 is not good. Question 4 here is talking about possibility consequences of poly A tail if it's shortened or deleted, and I chose these as my answers, and I said because it would be unstable because the 5 cap would be deleted or shortened, which causes the mRNA to degrade. mRNA will be less stable, which will impair both functions of localized mRNA, and transition will be blocked. By that being blocked, it causes improper localization of mRNA because it's shortened or deleted. So the blockage causes a lot more problems than we want. The secondary structure will not be formed because it is shortened or deleted, which means that it affects the mRNA localization. Sorry. And then for question 5 here, it's talking about how what would be the meaning why maternal mRNA is expressed for a longer period of time. And these are the reasons I'm saying is because 3 UTR in maternal mRNA is missing or mutated. Overexpression of histones in the fertilized cell, zygotic transcription is silenced, or microRNAs are unable to target the maternal mRNA because it is being expressed for a long period of time because it's missing so many other factors. These are the reasons that I would say that it's missing. A lot of it are either mutated, overexpressed, or it's silenced, or it's missing a certain protein. So that way mRNA doesn't know where to go or where to stop. So it just keeps getting expressed until that protein is placed back. Right here is talking about upregulation and downregulated. So we learned in class that CD9 and ZEMO play a lock and key. So CD9 is the receptor that ZEMO binds to. So if CD9 was to be regulated, we would expect an increase of the concentration of CD9. And if ZEMO was downregulated, it would kind of be the same thing. It would be decreased also. So I think that they're very much interchangeable. So if ZEMO downregulates on the sperm head, fertilization will be affected. So that means that less percentage of the change of ZEMO protein would be able to bind with the CD9 receptor. So if this occurs, then the male would be less fertilized. They're not infertile, but there's a lesser chance for it to, since it's downregulated, there's a lesser chance that it would be upregulated. So CD9 is a protein found in eggs, and ZEMO is a protein expressed in sperm and is hemoglobin. CD9 is needed for the sperm, and also because it binds with also the ZEMO, they need them both in order to have the receptors on the egg, which then allows the sperm to bind it. So if one is downregulated, upregulated, or something, they're not going to be able to combine with each other. And then question 7 right here, we're talking about mutations. I actually want to change my answer and say I would mutate 7 and 10. Like if you look in the figures 10 to F, I'm sorry, D to F, we would see the delocalization of the mRNA, but also 6 and 7 are grouped together. So I would like to mutate 1 and 2, but I want to do it separately to see if any change will occur. And if you see delocalization with 1 or 2, we could confirm that the site 6 doesn't control all of localization, because in each of these figures, 6 is also mutated, but 1 and 2 also is, but I want to be able to see them separately. Okay. And then for question 8 right here, we're talking about a female contraceptive. So I kind of took the birth control and make it a little bit of my own. So I'm saying in a pill form because it's a lot easier for women to take this pill daily because they most likely will keep it with them at all times. I believe this will work because it's easy. It targets the proteins in the female gametes. I believe that this protein will target ZP1, ZP2, ZP3, and target the CD9 receptor. So CD9 receptor is targeted because it's a protein that is responsible for the sperm binding to the egg, like we talked about in previous problems. ZP2 would be targeted because, like CD9, it is sperm binding and also used for polyspermine blockage. So this receptor will block those two proteins from binding the egg, which then the sperm and the egg. So then without those two, there would be no activation and no pregnancy will occur. That way, this pill will work. Okay, and then last one here for question 9, we're going to be talking about how the difference between autonomous and conditional. So This autonomous is for the cell differentiation on its own, while conditional, like the word condition, it has a lot of conditions in order for it to differentiate. So it relies on external signals in order to help start the differentiation. So example of autonomous would be stem cells. So they do it on their own. They don't need any other external factors, while conditional needs external factors. So conditional would be cardiac, brain, and liver. So an experiment that we learned in class that I want to use is cell to cell. So we're going to use a controlled experiment. Controlled to me would be more like conditional. So if it's autonomous, then it would not be a cell to cell. It doesn't need anything else in order for it to differentiate. But if we do something like conditional, if they interact with each other from one cell to another cell to differentiate, then we can see that it needs each other, and if it needs each other, then it would be conditional. So then we can set it and track each time that it moves, each cell, and then see what variable comes into play. And if it's autonomous, then you can just see that it just does on its own. It doesn't need any other factors inside there. And conditional, it would need each other in order to differentiate. Okay, so that is all that I have for you today. Thank you so much.